I first heard of Professor Kurup when my cousin Padma Tampati joined his department for the MSc course in biochemistry. And during the same time, I also became acquainted with Professor Venugopal Menon and Professor Sudhakaran, who were pursuing their doctoral degree. I was a medical student then. But then 2 decades elapsed before I met Professor Kurup. And one day, he walked into my office at Chitra unannounced, and I jumped up from my seat in wonder and respect. He had come to ask for tissue samples of heart with endomyocardial fibrosis to analyze their glycosaminoglycans compositions. He has published his studies on the heart in Indian Heart Journal. I have admired Professor Kurup for his sustained dedication for academic pursuits and interest in a broad range of subjects, and for actively engaging in research until his eternal exit from our midst. From his first paper in the Journal of Indian Institute of Science on his PhD work on the antibiotic principle from Moringa pterygosperma, he has amazingly a large number of publications, which are all available in PubMed. I'm sure there will be more publications which are probably not listed in PubMed. And 54 publications came in 2003 when he was almost 76 years old. And 1 year later came his most cited work in Indian Journal of Medical Research. He has shown interest in a wide range of disciplines, whether it is vascular biology, nutrition, toxicology, neurosciences, ethnopharmacology, and of late in evolutionary biology as well. I have chosen for today's lecture, in tribute to Professor Kurup, a topic possibly dear to him as well as to me, and that is an ancient disease. Atherosclerosis, though an ancient disease, that it remains fascinating disease because its origin is yet unknown and its prevention continues to be challenging. So in my lecture would have 3 parts. I would briefly review the significant contributions to understanding of atherosclerosis, atherogenesis, and its management. I would briefly dwell upon the modern prospects and also outline the atherosclerosis research done in my group. We all know that atherosclerotic lesions were discovered in Egyptian mummies dating from 1580 BC. Anatomists called it bone in the heart, or a bony aorta, or wounds of the arterial system. And it was John Frederick Krull, a German physician, who recognized the nature of the lesions and the fatty matter in them. It was Bicket, a French pathologist, who stated that the initial lesions are actually in the intima of the large vessels. And Lobstein coined the term arteriosclerosis. Virchow proposed a mechanism for atherosclerosis, and he said that a deposition or imbibition of material from the bloodstream into the arterial intima and the alterations that followed were the mechanism for the lesions. And he noticed proliferation of connective tissue and increase in the ground substance in the vessel wall. But what is the cause of these lesions? The first insight on causation was from the experimental studies by the Russian scientist Ignatowski. He demonstrated that a high protein diet produces atherosclerosis in the aorta of rabbits, but actually his high protein diet was a mixture of milk and egg yolk. The modern era of atherosclerosis research began with Anishko. These are all classic studies which I'm sure you are all familiar with. He showed that simply by feeding rabbits, one can produce vascular lesions which closely resemble those of human atherosclerosis, both macroscopically as well as microscopically. And he made very significant observation. One, that the endothelium of the lesions was intact. Second, that the earliest lesions appeared at the root of the aorta and aortic arch. Third, there were characteristic patterns of the distribution of early lesion, which he attributed possibly because of certain hemodynamic factors. And 4, there was ultimately deposition of connective tissue, which thickens the lesions in the vessel wall. And you also observed that the early lesions are reversible, but if it is the lesions continue for a long time and the fibrous tissue accumulates, then these are not reversible. And the extent of the lesion was proportional to the degree of blood cholesterol increase. Cholesterol was known at that time as cholesterol, and he said probably that was entering the arterial wall from the blood. And cholesterol-laden cells were probably white blood cells that had infiltrated the arterial wall. Anishko's findings were not taken seriously because the blood cholesterol levels in his rabbits were extraordinarily high, and people said that these kinds of levels are not seen in humans. But soon after his original studies, Anishko also showed that more modest increases of cholesterol levels were sufficient to induce lesions, only that it would take a longer time. It took more than 30 years to appreciate Anishko's contribution to settle the control controversy. His findings were largely rejected, they were not followed up, and most physicians considered that atherosclerosis was an inevitable accompaniment of aging, and nothing could be done, and nothing was being done. During the first 2 decades of the 20th century, a number of investigators had drawn attention to the fact that a considerable amounts of lipid in serum may be present either in some sort of emulsion or in association with proteins. Machibov and colleagues, they started the first definitive studies on the nature of the lipoproteins, plasma lipoproteins, which carry cholesterol. Russ and colleagues made an important finding that women before menopause had consistently higher blood levels of lipoprotein than did men. And they speculated that this difference could be the reason for the lower incidence of coronary artery disease in premenopausal women. Their paper was possibly the first demonstration that different lipoprotein classes might have distinct biological function. And probably this was the first suggestion of a linkage between specific lipoprotein patterns and the risk of coronary artery disease. But their speculation was at that time based only on a correlation. They could not establish a causal relationship. An important milestone in atherosclerosis research was a shift in emphasis from cholesterol to lipoproteins. And thanks to John F. Goffman and co-workers at the Donner Laboratory at the University of California. Goffman discovered and described 3 major classes of lipoproteins. And their work actually revealed the nature of the giant molecules of the serum, and that it composed of cholesterol, phospholipids, fatty acids, and protein as building blocks. Goffman had actually accepted Anishko's dictum that cholesterol in the blood somehow played a causative role in atherosclerosis. His basic premise was that a defect in some of these giant molecules would be responsible for the development of atherosclerosis. The next 2 decades witnessed an explosive increase in research on plasma lipoproteins and its relationship to atherosclerosis. And we came to know more about the lipoprotein metabolism, the function of these lipoproteins, and their correlation with atherosclerosis. The development of analytical ultracentrifuges later led to the discovery of the considerable diversity of components in the low-density group. These molecules are present in increased amounts in the cholesterol-induced atherosclerosis in rabbit, as well as in patients with myocardial infarction. And there were other major discoveries that followed, which won Nobel Prizes for Conrad Bloch and Gossin and Brown. And there was discovery of patients with hypercholesterolemia and classification of the hypercholesterolemia and the subsequent discovery of the LDL receptors. The case against hypercholesterolemia became stronger based on pathology, clinical observations, genetic studies, and epidemiology. And clinical interventions with trials with diet suggested that one can reduce the hypercholesterolemia and thus reduce the coronary artery disease risk. American Heart Association accepted the causal relationship and recommended that people at high risk should be advised to modify their diets. These dietary recommendations were extended to include the general public as well, not only the patients. And the similar recommendations are made by the Food and Nutrition Council of the American Medical Association. The epidemiologic evidence was provided by the 7-country study of Ansel Keys, the famous 7-country study. Their data revealed that the blood cholesterol levels are proportional to the saturated fat intake. And in fact, in Japan, it was only a little over 160 milligram per deciliter, whereas in East Finland, it was over 260. And proportionately, there was a difference in the incidence of fatal heart attacks. What was needed then was an intervention trial. And this was done. There were 3 landmark intervention trials: the Paul Laren Oslo study in 1966, the Finnish Mental Hospital study in 1968, and the Wadsworth Veterans Administration Hospital study in 1969. And all of them indicated that the diets rich in polyunsaturated fat would significantly lower serum cholesterol levels. And the studies collectively provided a very strong evidence for the lipid hypothesis. But still, the idea that the cholesterol level could be centrally important was rejected, at times quite angrily, by many cardiologists and nutrition experts. In 1976, an editorial in the British Heart Journal concluded, I quote, "The view that raised plasma cholesterol is per se a cause of coronary heart disease is untenable." And Michael Oliver, who has been dubbed as a cholesterol pessimist, wrote in 1981, I quote, "It is probably of little value to reduce raised serum cholesterol levels in patients with overt coronary heart disease." Interestingly, Oliver's skepticism was shared by many others as well. Then came the Framingham Heart Study. The Framingham Heart Study provided the first solid and unarguable evidence that individuals with high blood cholesterol levels were more likely to experience a heart attack. This study, which began in 1950, included 28,000 residents of Framingham. And the initial cadre was followed with periodic examinations for more than 24 years. The final evidence for causation was provided by the Coronary Primary Prevention Trial. This is a large randomized double-blind study which showed a statistically significant decrease in heart cardiovascular endpoints as a result of decreasing cholesterol level with the use of a bile acid binding resin, cholestyramine. Professor P.A. Kurup's impressive forays into atherosclerosis research was during this period. And he look at the large number of themes on which he and his students worked. The role of dietary fiber and dietary starch on cholesterol metabolism, role of cigarette smoke and nicotine on cholesterol metabolism, hypolipidemic effects of black gram, red gram, horse gram, plantain, coconut kernel, tubers, and paddy, metabolism of glycosaminoglycans in atherosclerosis and diabetes, effects of magnesium deficiency, zinc, copper deficiency, vitamin A, ascorbic acid, whole lot of nutritional elements, methionine, hormones, sardine, coconut, saffron oil. They are all the effects of all these on lipid and or glycosaminoglycan metabolism. Until the 1980s, there was still lack of an accepted detailed hypothesis for lesion development that mechanistically linked lipoproteins to the pathogenesis of atherosclerotic vascular disease. And this was provided by Russell Rose, another pathologist. He suggested that atherosclerosis developed as a response to the arterial wall to injury. And his proposal initiated a complete shift in atherosclerosis research. And there was focus on understanding the biology of vascular function and repair. His 2-part review, which was published in New England Journal of Medicine, quickly became the standard reference for pathogenesis of atherosclerosis. So the response to injury hypothesis states of 2 steps. One is an endothelial denudation of dysfunction, which could be caused by many factors: shear stress, elevated LDL levels, free radical, genetic alterations, increased plasma homocysteine, infections, or a combination of all these. And the step 2 is endothelial dysfunction, which leads to compensatory responses that alter the normal homeostatic properties of the endothelium. There is increased adhesions of the endothelium, formation of vasoactive molecules, procoagulant activity, increased permeability. And if the inflammatory response does not remove the offending agents, it would stimulate the migration and proliferation of smooth muscle cells, resulting in thickening of the wall, remodeling of the vessel wall, continuous inflammation, release of hydrolytic enzyme, further damage, and repeated cycles of enlargement of the lesion and occlusion of the ligament. And this picture must be very familiar to you, how oxidized LDL might be involved in atherogenesis. Then came a new era in the treatment of hypercholesterolemia, not just reducing cholesterol levels by dietary means, by reducing cholesterol levels by drugs. And that was the birth of the statins. Akira Endo in Japan speculated the broths in which fungi were being grown in the hunt to discover new and better penicillins, they may also contain natural inhibitors of cholesterol synthesis. He hoped that some microorganism might be producing these compounds to fight against other microbes, which required sterols or other isoprenoids for growth. 6,000 tests later, which took almost 2 years, he finally came up with a real winner. The cultured broth from Penicillium citrinium had a remarkably potent inhibitor of cholesterol synthesis, which he named compactin. And he found that if you give compactin at a dose of 50 milligrams per day, that would reduce cholesterol levels in patients with hypercholesterolemia by an average of 27%. And in some, it could reduce as much as by 30 to 35%. Then came the statin clinical trials, the famous Scandinavian simvastatin survival study, which found that the total cholesterol could be reduced by 25%, LDL cholesterol by 35%, and coronary heart disease deaths by 42%, a reduction which is highly significant. This was the study for the first time which showed a significant decrease in all-cause mortality by lowering cholesterol levels. Niacin was found to reduce high-density lipoprotein by 15 to 35% if given at high doses, and niacin is found in many foodstuffs. The discovery that plaque structure was the major cause of myocardial infarction and stroke demonstrated that new treatments should be aimed towards stabilizing plaques and preventing plaque rupture. Two people independently, Erling Falk in RS Denmark and Michael Davis in Cambridge, found that it was not the size of the plaque that determined how dangerous it was, but its vulnerability to rupture. And they showed that heart attacks were caused by ruptured plaques activating formation of a blood clot, completely occluding the coronary artery. Several pathways in atherosclerosis network have been delineated in subsequent years, whether it is the metabolic pathways, the LDLR pathway, the pathways of glycolipid metabolism or metabolic syndrome, inflammatory mechanisms, cytokine networks, signaling mechanisms, etc., etc. All these discoveries lead to discovery of new therapies for atherosclerosis. There was a new generation of therapeutic targets. And these targets were either lipoprotein metabolism, biologically active lipids, leukocyte recruitment and retention, or extracellular matrix turnover and plaque rupture. You can see the long list of agonists or inhibitors of antisense oligonucleotides or gene replacement therapies targeted at the lipoprotein metabolism, or biologically active lipids, or leukocyte recruitment and retention, or extracellular matrix turnover and plaque rupture. This list is almost endless. But the modern perspectives commence when people started thinking whether inflammation and hypercholesterolemia are partners in the crime. The progress in clinical and translational atherosclerosis research has brought inflammation in clinical focus. Inflammation is an established feature of all phases in atherosclerosis. This was recognized long ago. Disease initiation involves pro-inflammatory activation of endothelial cells and capture of circulating monocytes and T cells. The macrophages are involved, and even smooth muscles may contribute to contribute as a stabilizing pro-fibrotic factor. Inflammation also plays a critical role in the fatal complications of atherosclerosis. because plaques become unstable due to inflammation. They rupture, erode, and that could lead to atherothrombosis. Infection could be a cause for endothelial cells. People have speculated an aggregate pathogen burden of cytomegalovirus or CHESV plus chlamydia and hepatitis A virus. All this could cause inflammation and cause endothelial injury. But the current thinking is whether atherosclerosis is an autoimmune disease. If you look many standard textbooks, you would find atherosclerosis being classified along with autoimmune diseases like liver cirrhosis, rheumatoid arthritis, glomerulosclerosis, pulmonary fibrosis, and chronic pancreatitis. In 1989, scientists at University of California identified antibodies against oxidized LDL in human serum. And that was the first demonstration that there is a presence of an adaptive autoimmune response against oxidized LDL in man. This observation of the existence of an endogenous atheroprotective immune response directed against antigens present in oxidized LDL opened a new path towards developing completely new forms of prevention and treatment of cardiovascular disease. Could these protective immune responses be selectively activated using a vaccine approach? So these are the milestones in the path towards development of a vaccine to prevent atherosclerosis. Adaptive immunity and atherosclerosis was recognized in 1985. And 10 years later, it was demonstrated that LDL immunization could reduce atherosclerosis. And during 2005 to 2015, there was refining of the specificity of this antigenic antibody response. And by 2015, started testing of vaccines and antibody therapies in clinical studies. The first challenge in the development of an atherosclerosis vaccine was finding the right antigen. One possibility would be to use a complete oxidized LDL, but oxidized LDL is a complex structure and it's likely to contain a mixture of a large number of different and poorly defined antigens. So because of this, it was important to identify and characterize the structures in oxidized LDL that were targeted by the immune system. Joe-Wittstern and Wolff-Palinski and their co-workers isolated antibody-producing B lymphocytes from the spleens of atherosclerotic mice, and they produced hybridomas, and then they produced antibodies, and these antibodies were sequenced, and then they used a vaccine to immunize atherosclerosis susceptible mice, which developed high levels of IgM against oxidized phospholipids. And they could demonstrate that the development of atherosclerosis could be reduced by about 30%. Apolipoprotein B100 was suggested as a possible target for immune response by Nilsson and Shaw and their co-workers. And their strategy was entirely different. They knew that when LDL is oxidized, this large protein is broken down into smaller fragments, and then they get contaminated by the attachment of small and extremely reactive molecule called aldehydes. So the aldehyde modified peptide fragments of ApoB100 represented another possible target for immune responses against oxidized LDL. They analyzed the plasma from a large number of individuals, and they could identify antibodies against about 100 different ApoB100 peptide fragments. Then they investigated whether immune responses against any of these fragments could produce protective effects. And vaccine based on a number of different ApoB100 peptide fragments have been tested, and a handful of peptide vaccines that reduce the development of atherosclerosis by 50 to 60% have been identified. Based on the studies in mice immunized with oxidized LDL, Shaw and colleagues suggest that it should be possible to treat atherosclerosis by modulation of the immune system, and the concept of an atherosclerosis vaccine was later proposed by Hansen in 2001. A number of possible antigens for such vaccines have been identified and are proved to be effective in animal studies. I look at the large number of vaccine targets. CD99 target, and one could reduce atherosclerosis by 38 to 69%. VEGFR2 reduce progression of pre-existing advanced atherosclerotic lesions by 66%. PCSK9, it is being clinically used. PCSK9 specific monoclonal antibodies are being clinically used, and they, along with statins, can lower LDL levels approximately by 60%. Apolipoprotein immunization with the native antigens could reduce atherosclerosis by 59%. Heat shock proteins, beta-2 glycoprotein, cholesterol ester transfer protein. Though clinical trials using agents that inhibit CETP activity resulted in increased mortality, there is a recent revelation that you can use CETP antibodies along with statin, and that could reduce the risk for coronary events. Although these results show great promise, there is still a long way to go before a human atherosclerosis vaccine could become available. There are several challenges ahead. What are these challenges? One is to improve the preclinical models to test the efficacy of this therapeutic approach. Second, define the most optimal tolerogenic adjuvant in vaccine formulations for human use. Third, elucidating the yet unknown aspects of immune responses in atherosclerosis. Is there a single antigenic determinant driving the immune response? Or can we develop a widely applicable tolerogenic vaccine? Or whether we should follow a personalized approach, different anti-vaccine for different group of patients. Identifying the preferred target population for a tolerogenic vaccine, and identifying the endpoints for clinical trials. Should this strategy be aimed only to patients with high inflammatory risk or to the general population of patients with atherosclerosis? Can we design large-scale clinical trials with endpoints that rely on plaque morphology and immunotyping? How an atherosclerosis vaccine may influence any possible pre-existing protective immune response. So these are the challenges ahead for developing a vaccine. But there have been other new strategies being developed to fight cholesterol problems. And one is a targeting defective efferocytosis. Efferocytosis has been found to be defective in atherosclerotic lesions, and this is related to an upregulation of the do not eat me signal, the CD47. So mechanistic studies have demonstrated that the expression of CD47 and ultimately impaired efferocytosis is under the control of TNF-alpha. So anti-TNF-alpha treated patients, one could reduce the expression of CD47, and that could improve efferocytosis, and that will reduce the magnitude of the atherosclerotic lesions. So impaired mechanism that regulate macrophage death and removal, whether it is apoptosis, necroptosis, or efferocytosis, they do play a role in pathogenic. They do have a pathogenic role in atherosclerosis, and hence they're also novel therapeutic targets. The other type of developments in nanotechnology, developing nanoparticles of high-density lipoprotein, which could be used to improve the levels of high-density lipoprotein in patients. Another major area of advance in atherosclerosis research is looking at the genetic susceptibility of atherosclerosis. We know that several monogenic diseases elevate plasma levels of LDL, but the majority of atherosclerotic lesions are not due to monogenetic heredity, but they have several major genes are involved in the manifestation of the disease. And polymorphism within genes in lipid metabolism, inflammation, and thrombogenesis are probably responsible for a wide range of atherosclerotic disease. And genome-wide association studies and experimental studies have revealed the genetic background of LDL oxidation in the vessel wall, the initiation and the formation of an early fatty stick lesion, and a series of lipid-modifying enzymes in the vessel wall. And also the basis for genesis of inflammatory mediators. I'll conclude by citing some of the work done in my group, and our focus has been on inflammation and atherosclerosis in type 2 diabetes with the aim of identification of therapeutic targets and biomarkers. While I was in Sree Tirunal Institute, Sumit Panikar asked this question: can monocyte chemoattractive protein 1 be a target to reduce the increased risk for atherosclerosis in type 2 diabetes mellitus. We all know that MCP1 is synthesized by activated endothelial cells. It's a chemoattractant, and it has been implicated in the pathogenesis of atherosclerosis. Inflammation is mediated by MCP1. It can accelerate atherosclerosis, plaque destabilization, thrombus formation, and restenosis. And it could work as a positive feedback loop for increasing the cytokines and chemokines, addition molecules, growth factors, MMPs, and tissue factor. It has been shown that MCP1 accelerates atherosclerosis in apolipoprotein E deficient mice, and MCP1 deficiency reduces the susceptibility to atherosclerosis in mice that overexpress human lipoprotein P. So the objectives of his study was to find whether MCP1 gene expression is a key step for the increased risk for atherosclerosis in type 2 diabetes, and whether MCP1 could be a target to reduce the increased risk for atherosclerosis in type 2 diabetes. And his studies demonstrated that there are specific targets in the signaling pathways involved in the MCP1 expression induced by high glucose in aortic endothelial cells, and that the increased MCP1 expression in endothelials can be attenuated by using substances which block these sites in the signaling pathway. He found that quercetin and curcumin significantly attenuates high glucose-induced MCP1 mRNA and protein synthesis, as well as they prevent reactive oxygen species generation by high glucose. Surya Ramachandran, my colleague at Rajiv Gandhi Center for Biotechnology, asked this question: what causes the acceleration of atherosclerosis in diabetes? Is it possible to detect the progression of vascular disease in diabetes using a serological marker? Her focus was on monocyte because monocytes are central in atherogenesis, and it's also known that it initiates lesion formation in atherosclerosis and also associated with lesion progression in atherosclerosis. And hyperglycemia in type 2 diabetes can activate monocytes. She demonstrated through proteomic profiling of high glucose prime monocytes that cyclophilin A, an immunophilin, is secreted by these monocytes which are primed with high glucose. She also demonstrated that the plasma level of cyclophilin A is increased in patients with type 2 diabetes mellitus, which suggested so this could be a marker to suggest a presence of vascular disease in type 2 diabetes patients. So cyclophilin levels are associated with HbA1c, fasting blood sugar, and age. Cyclophilin levels were higher in patients with diabetes mellitus and coronary artery disease, and it was positively associated with the levels of C-reactive protein. Then she looked at the mechanism for accelerated macrovascular disease in diabetes mellitus, the cyclophilin A-induced mechanism, cellular mechanisms, and found that cyclophilin A enhances macrophage differentiation and lipid uptake in high glucose conditions. Cyclophilin A increased monocyte adhesion and transmigration into the endothelium under high glucose conditions. Cyclophilin A promoted monocyte to macrophage differentiation. Cyclophilin A induced oxidized LDL uptake and stimulated monocyte-derived macrophage form cell formation in vitro. Cyclophilin A induced form cell formation in human monocytes as well. It could induce oxidative stress during form cell formation in hypoglycemic conditions. Which all summed up, one could say that cyclophilin A promotes vascular inflammation in hypoglycemia. The extracellular cyclophilin could induce addition of monocytes to the endothelium, and intracellular cyclophilin could increase monocyte macrophage differentiation and form cell formation. Surya also showed that metformin, a common drug used in patients with diabetes, could attenuate the effects of cyclophilin on macrophages. Metformin decreased the transmigration of monocytes induced by cyclophilin A and high glucose. Metformin also reduced monocyte to macrophage differentiation. Metformin reduced form cell formation. Metformin reduced redox activity, and metformin also reduced TNF-alpha, MCP1, and cyclophilin A levels in monocytes. All this metformin did by repressing the cyclophilin A-induced inactivation of AMPK1 alpha in macrophages during lipid uptake. Vinita followed these studies by asking the question, does cyclophilin cause progression of atherosclerosis lesions in vivo? She has a rabbit model, a high-fat diet-fed rabbit model of atherosclerosis, and she found that cyclophilin A induces macrophage apoptosis and enhances atherosclerotic lesions in these rabbits. Cyclophilin A expression was found to be increased in the intima and medial cells of the aorta of the high-fat diet-fed rabbits. And intracellular cyclophilin A increased mitochondrial reactive oxidation species, resulting in both mitochondrial membrane potential loss and MTP opening, which all led to the release of cytochrome C and activation of caspase 3. Intracellular cyclophilin also induced chromosomal DNA fragmentation. So both the nuclease activity of cyclophilin A and induction of mitochondria-mediated death signaling pathways induced macrophage apoptosis. She has also shown recently that cyclophilin A can impair efferocytosis by overexpressing CD47 and downregulating calreticulin. My colleagues Sumi at Rajiv Gandhi Center Biotechnology did some genetic studies in patients with vascular disease, coronary artery disease, and diabetes. And she has demonstrated that ENPP functional variant, a functional variant of ENPP1 enhances susceptibility to coronary artery disease in South Indian patients with type 2 diabetes. And a non-synonymous gene variant of CX3CR1 in this same population is associated with a reduced risk for vascular disease in patients with diabetes mellitus. So this was a candidate gene approach to understand the genetic susceptibility for vascular disease in patients with atherosclerosis. So to summarize my talk, I would say that not many subjects in medical research are more challenging than atherosclerosis. Many different approaches have been used to understand the mechanisms involved in atherogenesis and devising the strategies for its prevention and its treatment. Many of them have been fruitful and rewarding as well. From Aristotle's description of bone in the heart to the experimental production of atherosclerosis in animals by Russians, and to the discovery of cholesterol biosynthesis and vaccines against atherosclerosis, the progress has been indeed a crescendo. So what is the future? I think the future would be discovery of more vaccines, and more possibly specific vaccines for against atherosclerosis for a personalized approach, and probably advance in precision medicine by identifying the genetic susceptibility for atherosclerosis and its clinical complications, leading to early detection and prevention. I think if Professor P.A. Kurup was alive today, he would be dabbling in these areas, because I consider he was a man who moved with the times. So I thank you all for a patient hearing. And once again, I thank Professor Helen and other organizers of this meeting for inviting me to give this lecture in honor of Professor P.A. Thank you very much.